Okay, good afternoon, everybody. We got a session here on gene therapy, gene editing, and I don't need that up. I've only got a few minutes. So we're going to cover quite a bit today. There's a session on gene therapy where we're having each of the leading microdystrophin companies come and talk about where the program is. We've got gene editing, and I think for the first time, Precision Biosciences is going to be here, talking about a permanent edit of the DNA gene, and with that trial, it'll start recruiting this year. Following that, I think for the first time, we've got Tavard Biosciences that's going to be talking around some work with tRNAs to read through nonsense mutations. And then Barry's going to come up and, I think, talk about safety and consensus guidelines for gene therapy. I'm not sure, because he keeps changing his mind on what he's going to do, but we'll see how we go. I'm going to spend a few minutes just giving a general overview of the gene therapy platform so that the companies who've only got about 8 minutes or 9 minutes each don't have to repeat that there. And we can talk about what gene therapy is and what it is not. This slide just really starts to walk into it. And gene therapy is fairly simple in Duchenne. It's a monogenetic disease. It's well suited to putting a single gene back in again, either a portion of a gene or a full one. And we can't just, in Duchenne, inject dystrophin back into muscle. Unfortunately, that doesn't happen. We need to get that DNA into all muscle. We need to get it into the nucleus. And the preferred way of doing that is very simple. We just adapt what nature has, and that there is use of virus. Nature has designed these viruses over billions of years to be incredibly efficient at delivering genetic material in the nucleus of cells. And the ones that we use is called an AAV, an adeno-associated virus. The reason for that is that it causes no disease in humans, and it's quite prevalent. And it's also got the ability to target a lot of different tissues. The benefits on the slide you can see is single dose, high transduction, broad specificity for different tissues, low immunogenicity, and long-lasting therapeutic benefits. But the challenges are the dose that we require, no ability to redose and limited cargo capacity. Now, viruses range from the very small to the very large. Some you can see under a microscope, some you'll need an electron microscope to see. An AAV is one of the very small ones. It's about, I think, about 30 nanometers in diameter. Doesn't mean a lot to you, but the diameter of a human hair is about 100,000 nanometers. So it tells you how small this virus is. It's one of the smaller ones. And that gives you clues into the carrying capacity is very limited. And as you know, the dystrophin gene is one of the largest genes that we have, and it is the largest gene in the human genome. So that presents challenges in trying to put the full-length gene in there. Now, we got around that by, again, adapting from nature. This here shows, the cartoon actually shows a cartoon of full-length dystrophin, and one that is heavily truncated. And that heavily truncated version actually came from an individual that was identified a number of years ago that has Becker muscular dystrophy. This particular individual walked into the clinic with the aid of a cane, age 61. He actually had a sibling, another family member with the same mutation that was a weightlifter, a bodybuilder in his 20s. So it gives you a sense that you can make heavily truncated versions of this gene and still have high functionality in muscle. And this here is still too much to put into the carrying capacity of an AAV. The coding region for dystrophin is about 14 kb. The carrying capacity of an AAV is under 5. So the truncation that's needed is even more than we've seen from this one in the human who's lost almost the whole middle of the gene. This particular individual had missing exons 17 through 48 and shortened it almost by half, but still too much to carry. And this here's been the template that each of the companies have worked off in order to design even shorter versions that we're using in clinical trials right now today. So each of the companies, we're looking through the drug product that they've got. It's comprised of a number of components. There is the capsid, which is the AAV, and each of the companies are using a different variation on them, from RH74 at Sarepta to AAV9, or a modified AAV9 at Solid, and AAV9 is being used at Insmed, and AAV8 is being used with REGENXBIO. So 4 different capsids depend upon what companies are wanting to do. There's the promoter. These Viruses deliver into many different cell types, but we only want the gene turned on in muscle. So the promoter is a muscle-specific promoter that only turns the gene on when it's present in muscle. The gene is not presented anywhere else. And then obviously the microdystrophin gene. So each of these are variations on a theme. There's different versions of the microgene that's been designed by the companies, different promoters, different capsids. So I view these as independent of each other with multiple shots on goal, and that's what we're seeing in each of these. So today, we're going to see updates from Sarepta, and this will be in about 3 or 4 minutes' time. I'll invite the first speaker up. REGENXBIO, who've just presented the top-line data about a week ago. Really exciting there to see that there come through. From Solid Biosciences, who've got a heavily modified capsid that actually makes their AAV more targeted towards muscle and less targeted towards the liver. And then Insmed, who are recruiting very young patients right now in their phase 1 ASCEND study. We're going to hear from each of the companies. Now I'm going to shift gears because I've got one slide to talk and introduce you to what's going on with Precision. It's the first time the company's been here, and their solution into Duchenne is quite different from what you've seen on delivering a microform of the gene. They're using technology in order to splice or cut the DNA with a one-time permanent cut. Their Arcuous genome editing tool is very small. In fact, it's small enough that 2 copies can be delivered inside a single AAV, and that becomes important for what they want to do. They want to cut at exon 45 and at exon 55, and you might say, why on earth would somebody want to do that? Well, there's thousands and thousands of mutations that are known to happen across disease-causing mutations across the gene, but they tend to cluster. And one of the main clustering regions is between exon 45 and exon 55. It accommodates roughly about 60% of all disease-causing mutations. And so a treatment that can target that region could actually effectively treat up to 60% of all patients with a single shot. And that's what the company's trying to do. The second component of it is that their excision of those 11 exons from 45 to 55 is a known and well-identified Becker muscular dystrophy phenotype. It's very mild, it's almost asymptomatic, so using that there as an approach to do a permanent cut and gene edit in there is an attractive approach, comparative to what we're doing with various other technologies out there. So they've got a very unique approach into this, and I think it's going to certainly catch the imagination of the community as they start to recruit their first patients this year. And then this one is quite a different one altogether. This is neither gene editing, nor is it gene delivery. This is Tavard Biosciences. And we first came across this work way back in 2023. I heard Deborah talk about it this morning. And we're immediately struck by the potential for what this is capable of doing. This approach takes over from what we wanted to do, what we were hoping to do with Translarna. Like, we were really disappointed that Translarna couldn't get approved in either Europe and the United States. And that left a very significant number of patients who have got nonsense mutations, somewhere around about 12% of all Duchenne individuals out there. And the importance of this here approach for us is that no one was going to be left behind. You know, that their mutation cannot be addressed by other ways. And so the work that Tavard was doing became really important in what we were thinking about for that strategy, trying to address the failings of Translarna. Now, their approach is, again, it's got a common theme all the way through this, is delivering tRNAs using an AAV. Their tRNAs are highly modified so that whenever it touches the nonsense mutation, it actually allows the cellular machinery to read through what is that their temporary stop codon. And the benefits are that you produce natural full-length dystrophin. And you're going to hear some data from the team that they've continued to improve the form of their suppressor tRNAs. They're highly efficient at what they're doing. They've got activity in vivo in the MDX mouse model. And you'll hear some work that I think puts us in a position where we've got real visibility now to that our program being able to move forward and get into the clinic in a shorter period of time. And the last one is just to remind me who's talking. I'm not going to go through this list, but I will invite up Wendy as our first speaker, and she's going to kick it off with an update on ELEVIDYS. Wendy, you're welcome.
Thanks, everyone. It's so great to be here. For those of you that we get to see year over year, this is such a special time, and thank you for bringing your families and meeting with us and getting to know us. And for those of you, this is your first year, what an experience this meeting is. As Mike said, I lead patient affairs at Sarepta. I'm going to do a little bit of a different presentation today in that I'm going to share a video. It's really hard in just a few minutes to take the journey of decades of research for gene therapy in Duchenne and condense it into time where we can share meaningful data and information with you. So we're trying a different approach. I hope you like it. I'd love to hear after this what your thoughts are. But this session's going to focus on data that's recently been released for ELEVIDYS in the EMBARK study, specifically 3-year data. This presentation is meant to provide education and scientific information, and it's in response to a request from the organization. Some of the information in this presentation includes data which come from an exploratory analysis. What does that mean? It means the results are early observations intended to help identify possible patterns or questions for future research. The analysis is not designed to provide definitive conclusions about treatment effects, safety, or outcomes. What's most important is to please talk to your doctor if you have any questions.
Only FDA-approved gene therapy indicated for the treatment of patients 4 years of age and older with Duchenne muscular dystrophy who have a confirmed mutation in the DMD gene. The prescribing information for ELEVIDYS importantly includes a boxed warning, which is the FDA's highest level medication safety warning. It is important to make physicians and families aware of possible major risks such as severe side effects. A boxed warning tells patients and healthcare providers about major safety risks so they can use the medication carefully and know what problems to watch for. The ELEVIDYS prescribing information also includes specific updated monitoring for liver function. Liver toxicity is a well-known risk across all AAV-based gene therapies, including those for Duchenne. Liver monitoring is very important, and urgent consultation with a specialist if acute serious liver injury or impending acute liver failure is suspected. ELEVIDYS safety is able to be monitored, with adverse events usually occurring within the first 90 days after the infusion. The most common adverse reactions, with an incidence of 5% or greater reported in clinical studies, were vomiting, nausea, liver injury, pyrexia or fever and high temperature, thrombocytopenia, which is low level of platelets in the blood, and increased troponin, which is a protein found in the cardiac muscle. In this video, we will share results from one of ELEVIDYS studies called EMBARK, specifically the 3-year data. In this study, no new safety concerns were observed between year 2 and year 3. We are committed to transparently share all data and learnings available with the community. Healthcare teams can monitor patients closely for any side effects so they can act appropriately and fast.
Patient Engagement Scientific Summaries, or PES, is Sarepta's innovative, differentiated capability designed for this moment when rare disease communities expect, demand, and deserve clear, plain-spoken scientific exchange. Duchenne muscular dystrophy is a severe progressive genetic disorder characterized by muscle degeneration and weakness, primarily affecting boys. It is caused by a genetic change, preventing the production of functional dystrophin, a protein crucial for muscle function. With every movement, muscles sustain irreversible damage, with damaged muscle being replaced by fat and scar tissue. As Duchenne progresses, it leads to early loss of the ability to walk, as well as respiratory and cardiac failure, ultimately reducing life expectancy. The impact on families is profound, affecting every aspect of daily life, physically, emotionally, socially, and financially. To better understand the safety and effectiveness of ELEVIDYS, researchers launched EMBARK, a Phase 3, double-blind, placebo-controlled crossover study enrolling ambulatory patients with Duchenne aged 4 to less than 8. ELEVIDYS is a single administration gene therapy that delivers a shortened, functional dystrophin gene to muscle. It is FDA approved in the United States. In Part 1 of this study, eligible patients were randomly assigned to one of two treatment groups, with one group receiving a placebo and the other group receiving gene therapy. This allowed researchers to compare treatment effectiveness versus natural disease progression. Part 2 of the study began after 1 year, at which time the patients who received placebo in Part 1 received gene therapy. 64 boys received gene therapy in Part 1. 52 boys continued from EMBARK in a long-term extension study called EXPEDITION. Before looking at the study outcomes, it helps to understand how ELEVIDYS is designed to work. ELEVIDYS uses a modified, non-infectious viral carrier. Once inside muscle cells, this gene aims to produce a protein that helps protect muscles from damage, with the goal to slow and stabilize disease progression. To evaluate the safety and efficacy of ELEVIDYS in eligible ambulatory patients with Duchenne, aged 4 to 8 years in patients, the study followed a defined treatment protocol. All patients in the EMBARK study began the process with a pre-dose of corticosteroids, 1 mg/kg per day, the day before treatment. The ELEVIDYS or placebo treatment was then delivered as a one-time, single-dose intravenous infusion. Post-infusion, patients continued corticosteroids for at least 60 days, in addition to their baseline steroid regimen, to help manage immune responses. A rigorous follow-up with each patient was conducted by the study team and adjustment made as clinically required. With treatment administered, functional endpoint assessments were monitored, designed to measure disease progression. The EMBARK study evaluates muscle function using 3 key clinical measures. The North Star Ambulatory Assessment, NSAA, the primary endpoint of a validated scale of overall motor ability, and 2 timed function tests, time to rise, TTR, and the 10-meter walk-run, 10 MWR, which assess speed and efficiency of movement. Together, these endpoints provide complementary insights into disease progression and the preservation of muscle function. And because gene therapies are intended to address the underlying cause of Duchenne and slow progression of the disease, these assessment measures become increasingly meaningful over time. Due to the wide variation in the progressive of disease in Duchenne patients, the cumulative impact of ELEVIDYS on cell function, tissue regeneration, or slowing disease progression may take years to fully manifest in clinical data. By year 2 follow-up, patients treated with a single dose of ELEVIDYS continued to show a slowing of DMD progression versus untreated peers. This group maintained or slightly improved on motor function, averaging a 2.63-point gain on the North Star assessment compared to untreated peers. As a result, the gap in muscle function between the two groups widened by 2.88 points. By year 3, the trajectory became even clearer. ELEVIDYS-treated patients, on average, remained above their baseline, maintaining function, while untreated participants declined. The functional advantage increased, reaching a 4.39-point difference. A similar pattern emerged while measuring everyday timed movements. Treated patients rose from the floor in just over 2 seconds, compared to untreated peers needing 8 seconds or longer. Their walking speed also doubled by year 3, a meaningful slowing of Duchenne progression measured in real-world movement. Importantly, the 3-year EMBARK safety profile remained consistent with the known and labeled risks of the therapy. The year 3 results demonstrate sustained, real-world functional preservation for the treated children and families. Beyond the study measures and safety monitoring, the meaning of these outcomes is profoundly personal. This explains the urgency to treat. Once a person with Duchenne loses muscle, they will never get it back. If you wait, you can never catch up. Every data point represents a child maintaining abilities, running, playing, living.
So every data point you heard in the EMBARK 3-year data for ELEVIDYS is actually a family that pioneered, committed to, contributed to, and saw through to the end clinical trial research, which helps all of us move the field forward. So when we say thank you to these families, we recognize it's time missed from school, it's missed family events. There's a lot of sacrifice that goes into joining and supporting a clinical trial. So thank you from the bottom of our hearts. We hope that this was a good way for you to hear this exciting data, and we're at the conference for the rest of the time, so happy to answer any questions when we see you. Thank you.
I'm going to go down there. It's my pleasure now to bring up the next one, Koji Takahashi from REGENXBIO.
Hi everybody. It's so great to be here today. Thank you to everybody in the audience, and thank you to the Cure Duchenne Foundation for such a great meeting as always, and for giving us an opportunity to speak today. As mentioned, my name is Koji Takahashi. I'm from REGENXBIO. It's a real honor today to talk about, to be able to present to you our pivotal data from the Affinity Duchenne study, which utilizes RGX-202, our investigational gene therapy for Duchenne muscular dystrophy. I'm going to try to get through a fair number of slides and data very quickly. But if you would like to learn more, I encourage you to check out the community webinar that Cure Duchenne hosted earlier this week with our excellent, most excellent clinical development lead, Dr. Naz Dastgir. So please take a look at that for more in-depth conversation. But without further ado, these are our forward-looking statements. At REGENXBIO, we've taken a very scientific and rational approach to designing the program around RGX-202. And it starts with our microdystrophin construct. So we're the only microdystrophin that encodes the C-terminal domain. And again, from a scientific approach, we felt that including the C-terminal domain would be really important for the function of this microdystrophin. And we've now shown in preclinical studies that including the C-terminal domain, as opposed to not including it, better preserves both skeletal and muscle health. We've also learned from others in the past in this gene therapy field, and we now know that the immune-mediated effects that occur as gene therapy is being administered really underlie the adverse events that we're all concerned about. And so we have designed a very proactive and short-course immunosuppression protocol to counteract these immune-mediated events, and we instituted it at the very beginning of our programs, and we haven't changed it one bit. The other thing that really, I think, differentiates us is we're very proud of the fact that we do all of our manufacturing in-house at our facility at Rockville, Maryland. Our proprietary methods allow us to develop capsids at a very high level, high purity levels, and we're also able to dose at a very high level of 2 × 10⁻⁴ genome copies per kilogram. And so these three really pillars that the foundation for this program we really hope leads to a differentiated approach in gene therapy. So our Affinity Duchenne pivotal study enrolled ambulatory boys greater than 1 year at screening. We included almost all gene mutations except for those with a mutation, point mutation or deletions in exons 8, 9, and or 10. Our primary endpoint was the proportion of patients with greater than 10% RGX-202 microdystrophin expression at week 12, and I'm happy to report today the 30 participants in this pivotal study, we met this endpoint with very high statistical significance. For secondary endpoints and functional measures for those greater than 4 years of age, we used velocity and time of the time function tests and NSAA. And again, for the first 9 participants that have reached 1 year in our study, happy to report the functional data today. And of course, safety is measured throughout our study. The demographics for this study, we enrolled 11 participants under the age of 4 and 20 participants greater than the age of 4, ranging from about 1.5 years to about 13.5 years of age, with the heaviest participant about 48 kilograms or 105 pounds. We are also reporting the safety data from this pivotal study, and overall, RGX-202 was very well tolerated. The treatment-related AEs were those that are commonly seen in gene therapies, like vomiting, fatigue, and nausea. But we also report for the first time 2 SAEs. One is a subacute myocarditis case, and the other is an asymptomatic liver injury. Just to go into a little bit of detail, the 8-year-old participant with a stop codon and exon 60 presented with mild abdominal and chest pain, normal troponin levels, but a slightly elevated high sensitivity troponin levels. The staff on site and the study investigators did an excellent job with both of these SAEs, and happy to report that this patient has resolved with no further sequelae. The liver injury as well was an asymptomatic liver injury, presented with elevation in GGT, which is a liver enzyme, just 2 times the upper limit of normal. This was a 10-year-old boy with a exon 3 to 7 duplication. Again, managed very effectively and by the study staff, and no further sequelae for this case as well. We also did not see any drug-related thrombocytopenia, myositis, or any neurotoxicity reported. As I mentioned, our primary endpoint was the biomarker of microdystrophin, and very happy to report that 28 out of 30 participants, or 93%, achieved greater than 10% microdystrophin expression at week 12. In fact, 80% of the participants had microdystrophin levels greater than 40%. And I know there's a lot of numbers and a lot of columns on this graph, but what this is basically telling us is that across the age groups, we're seeing really great levels of microdystrophin. And when we look at vector copy numbers, immunofluorescence, it's also telling us that it's in all the right places that we would want the microdystrophin to be expressed. Now, turning to our pivotal study, interim 1-year functional data. Again, this is from the first 9 participants who've reached 12 months of data. The Affinity Duchenne study is not a placebo-controlled study. We use external controls and multiple validated methodologies to look at the disease trajectory in the natural history cohort, who are obviously not on RGX-202. And we use that data to compare the functional data on those subjects that are in our clinical trials who were given RGX-202. This is a forest plot looking at various measures like the velocity of time to stand, velocity time to run walk, velocity time to climb, and the NSAA. And the values on the right side of the midline here are telling us that those on RGX-202 are showing improvements in all of these measures compared to the external control group at 1 year. When we look at those that were just 8 and over, which were 5 participants at the 1 year mark, in a very similar way, we're seeing improvements for those that were on RGX-202 compared to the external controls. And finally, for the first time, what we're showing is a correlation between microdystrophin expression and change in the NSAA. So just to walk through this figure really quickly, on the left-hand side is the change from baseline for NSAA scores on the y-axis there, and then on the x-axis is the change in microdystrophin expression. And so what we're showing now is a correlation that the greater the amount of microdystrophin being expressed, there's an improvement or an increase in the improvement in NSAA on the left-hand side. On the right-hand graph is the difference between NSAA and the expected disease trajectory in CTAP, and we're also seeing a correlation here as well. So just to summarize, we're really excited about this data. We have shown and we've demonstrated that we've met our primary endpoint of microdystrophin expression. We're seeing very robust levels of microdystrophin. We're very encouraged by the safety profile to date. The immunosuppression protocol that we've regimented and put in place, I think, is really demonstrating a great encouraging safety profile. Our interim functional data at 1 year is also demonstrating that we are exceeding external controls on both NSAA and all the time function tests. And we're really encouraged by what we're seeing in terms of the correlation between microdystrophin expression and change in the NSAA. With all of this data, we are planning to initiate a BLA submission later this year with a potential approval in the second half of next year in 2027. So with that, we would sincerely like to thank all of the participants and their families, the clinical trial staff and investigators, without whom none of this would be possible. And so from all of us at REGENXBIO, we'd like to thank all of our participants and thank you all in the audience for your attention.
Thank you, Koji. Thank you. That's real recent results for you guys. Next, we're sticking with gene therapy, and we're going to get an update from the team at Solid, where they're just beginning to recruit their phase 3 trial. So I welcome on Annie Gnott.
Thank you so much, Mike. Thank you so much, Deborah, Paul, Hawken, Liana, the entire team at Cure Duchenne. Truly, I am really, really grateful for you guys putting on this excellent conference every year. Of course, also thank you so much for the invitation to provide an update today on Solid's gene therapy program. We're very excited to be here. I'm Annie Gnott. For those of you that I haven't had the pleasure to meet, I am one of the co-founders of Solid Bio. I lead patient advocacy, and I am also a Duchenne mom. Forward-looking statements. We are a publicly traded company. I will make you memorize, and we can test you on that later. It's very exciting. All right, so Solid, we founded Solid in 2013 following our son's diagnosis of Duchenne. What started off as an extremely personal mission to try to improve the lives of all young boys and men living with Duchenne has turned into a company that is aggressively advancing multiple clinical programs across neuromuscular and cardiac. We have a long-standing relationship with advocacy organizations all over the globe for our indications that we are focused on. And most importantly, every decision we make is guided by our patients. They are truly our North Star in what we do. I go home to one every day, so I know how important it is to make sure that everything we do has the patients front and center. We're a lean company. We are 130 people based in Boston, Massachusetts, and we're really excited to be here today, so thanks so much. I'm going to dive into an update on our program. So just taking a brief step back on the science and explaining a little bit more about the unique nature of SGT-003, which is our next generation program for Duchenne. Mike did a wonderful job providing an overview of gene therapy. Specifically for this program, we're using an optimized transgene, or microdystrophin, which is, ours includes the nNOS binding domain. We've also put a significant amount of resource and effort into building out what we believe are next generation capsids, which are the delivery mechanism. For this program, we are using what is called SLB-101, which we have now cleverly nicknamed Polaris 101. So if you see that, that's what this is. So, and the goal of this new capsid is to ensure that we are making sure that the transgene is getting to the right place and getting to, of course, skeletal cardiac muscles and detargeting to the liver. And on top of that, also an improved manufacturing process, really focused on purity. Okay, so a little bit, I'm not going to spend too much time here, specifically for our microdystrophin, as Mike explained, that the Duchenne gene or the dystrophin gene is extremely large. We have the packaging limitations with respect to our AAV. So as he so beautifully described, academics over many, many years have figured out how to create these much more shortened length dystrophins or microdystrophins that are able to fit inside the AAV capsid. Specifically for our microdystrophin, we partnered with Jeff Chamberlain and Dongsheng Duan and came up with a newer transgene, which includes the R16, R17 in the middle, which is the nNOS binding domain. And the nNOS binding domain is really to try to help prevent activity-associated muscle injury, and ultimately to help preserve muscle function. I'm going to touch on both of our clinical trials that we have ongoing. The one on the left is Inspire Duchenne, which is our first-in-human phase 1/2 clinical trial. It's open label, all patients receive drug, one-time infusion. I'm also going to then, after I dive into that, share a bit of an update on Impact Duchenne, which is our phase 3 double-blind, placebo-controlled clinical trial of SGT-003. So I'll provide a study overview and some of the interim safety and efficacy results. Okay, so again, open label, every participant receives drug. It is a one-time infusion. We are, of course, safety is paramount. We're also looking at microdystrophin expression and a ton of functional assessments. For the Inspire clinical trial, we have 15 clinical sites across the United States, Canada, the UK, and Italy. We announced as of the 12th of May that we have dosed 47 participants in this first study. We are getting to the point of wrapping up enrollment in the first 3 cohorts. This clinical trial is enrolling participants that are between the ages of 0 to less than 12 years old. I'm going to dive a little bit into the safety that has been reported. We did report one SAE last year that was an immune-mediated myositis. Thankfully, was treated with steroids, improved. The patient is doing great. There's also a number of AEs that, as we've discussed, are associated with the field of gene therapy that include nausea, vomiting, decreased appetite, thrombocytopenia, abdominal pain. Our clinical trial utilizes our immune modulation. We do not use additional agents. We are using steroids only, and increase that tapers down. That's the orange box on the right-hand side. Okay, a little bit of what we're seeing in terms of results. The orange box on the left side, you'll see this is from a muscle biopsy. The top left is baseline, obviously no dystrophin expression at baseline. And then at that day 90, so that 3-month time point, you can see a cross-section of muscle, and the red staining is the muscle staining for our microdystrophin, for SGT-003. And then the middle box, this is what's encouraging to us, and what continues to make us excited for this program, is not only are we able to deliver a robust amount of microdystrophin expression, but on top of that, we're also seeing durability. And so those 3 sections of graphs are the different ways that we measure. So we measure it using Western blot, mass spec, and immunofluorescent staining. And you can see the dark blue is at that 90-day time point, and the lighter blue is the day 360. So not only are we getting robust expression, but we're also seeing durability. Then we're going to dive a little bit into markers of muscle integrity. So I'm sure for a lot of you, maybe around the time of diagnosis, you started to hear a lot more about CK. Certainly that was my introduction to CK. It is an enzyme that is released in the blood when there is muscle breakdown. You also have ALT, AST, which are typically associated with liver injury, but of course is also associated with muscle integrity, LDH, and titin. And everything is trending in the direction that we want to see. Ultimately, we are hopeful that this will ultimately result in a slower disease progression and better long-term outcomes. I'm going to now dive into Impact Duchenne, our phase 3. We have 2 clinical sites that are currently active and enrolling participants. We announced more recently that we have dosed our first participant in our phase 3 study in Australia. It is a double-blind, placebo-controlled study that is randomized 1-to-1. All participants will either receive SGT-003 or they will receive a placebo administered by infusion. And then there is an 18-month crossover. So for those that do receive placebo, at that 18-month time point, they would then cross over to receiving drug. One other thing on this, we are actively working to open up additional sites in the United States, the UK, and multiple countries in Europe. Who may participate? We are enrolling participants that are between the ages of 7 and less than 12 years old. We require patients to be ambulatory. We have exclusions based for mutations, and we can go into a little bit on that, but that's a longer conversation, and I'm happy to discuss it because I'm already past my time, so I apologize. We are excluding participants that have deletion mutations in exons 1 to 11, 42 to 45, or 57 to 69. They must be negative for AAV antibodies, no history of gene therapy, at least 6 months washout for exon skipping therapies, vamorolone or givinostat, and on a stable dose of daily oral steroids, prednisone or deflazacort for at least 6 months, and are not currently on any other investigational or approved Duchenne therapy. Endpoints, time to rise at that 18-month time point. Key secondaries, you've seen these all before. Stride velocity, 4-stair climb, North Star, and of course 10-meter walk run. And I'm sorry, I'm rushing now. Also, so we have here, we have 2 QR codes that take you to clinicaltrials.gov that has more information in terms of eligibility criteria, study sites, et cetera. So you have those QR codes there. We also include an email address if you'd like to reach out to us directly. Ultimately, I just want to share my profound thanks to all of the participating families, whether it's in a solid study or other. We know that it is nothing short of heroic efforts to participate in clinical research. So thank you, thank you, thank you. And if you have any questions, please come find me or my colleagues or come to our booth. So thank you so much. Thank you.
Thank you, Annie, and congrats on the progress. The final one of this particular part of gene delivery is Mark Stahl from Insmed. Insmed's secret sauce is their route of delivery. They've switched from intravenous to intrathecal, and that's learnings from other gene therapy products like Zolgensma. So to dig into that, I'll invite Mark onto the stage. Yeah.
All right, well, thanks, Mike, but you gave away the big reveal. My name is Mark Stahl, and I'm at Insmed Gene Therapy. I'm a neurologist and neuroscientist by background and training. And I just wanted to say a little bit of an introduction to Insmed for people who aren't familiar, since we haven't had as much of a presence here at this meeting until now. We are headquartered in New Jersey, and we're a company that really focuses quite a bit on serious and rare diseases for patients around the world, with a significant focus on folks who are kind of overlooked by sort of medicine in the mainstream. And we really try to put patients first and make a genuine effort to listen to them and incorporate their viewpoint in everything that we do. And I want to thank Cure Duchenne for inviting us to talk about our program, INS 1201 and the ASCEND study. So this is a diagram of INS 1201. It's an AAV9-based microdystrophin gene therapy for DMD. Our philosophy in designing this therapy was a little bit different from what you've heard so far. We really focused on taking the components that we thought would be the best available things that we could use that were already well characterized and well understood, and that we had high confidence that they would be safe and effective, and combining them into a single therapy. And then, as Mike pointed out, administer them in a unique way, which is, of course, to give them intrathecally instead of intravenously. So that is into the spinal fluid instead of into the vein. And why would we do that? That sounds a little bit counterintuitive, right? We're trying to get our drug into the muscle, so why would we give it into the spinal fluid? And maybe a little bit of preclinical data could convince you of why we think this was a good idea. So here you can see an experiment that we did in monkeys. And there are two groups. At the top is the exact same dose of the exact same AAV9 with a marker gene that's delivered intravenously into the animal's vein. At the bottom, you can see the exact same doses, so the same amount of drug, of a virus, given intrathecally. And I don't need to put any numbers on it to convince you, I don't think, that the efficiency of that expression, even at the exact same dose, is higher in the muscles that received their dose intrathecally versus intravenously. And you can see that holds true for all of the muscles that we tested, including the muscle of the heart. But what's even more exciting than that in some ways is that in addition to having that very nice delivery and expression in muscle, we avoid having nearly as high expression in the liver. And so as you know well from some of the other discussions that we've had, we intentionally try to avoid having too much virus in the liver because we think it can be a safety risk. And so the combination of having more expression in muscle, very efficient, and less expression in the liver seems like a win-win. And this is really the underlying basis for why we think our administration into the spinal fluid is really advantageous for how we give our drug. And that leads me right into the clinical trial that's already ongoing that we call ASCEND. ASCEND is a phase 1 study. And so by that, of course, I mean that we're focused on safety. And it's the first in humans and the first time that really anyone has tried to give a gene therapy into the spinal fluid and have it have an effect in the muscle. And so we're learning quite a bit with this first in human study. It's an open label study that doesn't have a placebo arm, and it's enrolling boys who are 2, 3, and 4 years of age. And as I mentioned, of course, our primary point in doing the study is to look at safety. We'll have an initial period of the study that's about 2 years, and then we'll move into a long-term safety study for a total of 5 years. And the main thing that we're trying to learn, other than, of course, answering the question about how well tolerated the drug is to really understand our dosing, since that's the thing about the treatment that's unique. And so ultimately, as we'd like to treat additional patients, we need to understand how the administration into the intrathecal space works. And so coming up with our recommended doses going forward. And we'll be doing that in large part by looking at how much of the microdystrophin is expressed via muscle biopsies. A few other things, of course, we're always interested in how our patients are doing, and we'll be using many of the tests that are frequently done in other DMD studies, but also because we are focused a little bit more in our first study, at least, in younger patients, we have a few things that we like to highlight about our study that both focus a little bit on things that we think are relevant to some of the younger boys who are participating in our study, but also reduce the burden on families to the extent that we can, because it's always a focus of our company to try to reduce the burden on families as much as possible. And so we have a couple of our assessments that are able to be done in the home. And so you might be familiar with the SV95C, which is collected via a wearable device You can see on the ankles of the boy in this first picture here. And so all you really have to do is convince them not to take it off and let them wear it for a little while, and we'll be able to collect our data. And then we also do some collection of videos in the home environment. Again, saving a trip in to the clinic to collect some important data that we think is quite relevant. And then on the sort of relevant side, we also look at developmental milestones. And so we see that how we can improve those so that they are closer to how kids who don't have DMD develop. These measurements, we feel, are part of the whole way of looking at the effect of our drug, but again, emphasize that patient-centeredness that we believe in strongly. This is a map showing some of actually all of our clinical sites that we have to date. They're all in the United States at this point, and I hope if you're interested in our study, there's one that's near you. But this lets me bring up another point about our patient-centeredness, which is that if you're not near a study site, we have a service, a concierge service that will help you with travel arrangements and get you where you need to be. And we take put a lot of effort into making that simple and easy. On the right side, you can see a QR code and some ways to get in touch with us if you have interest in the study. So in summary, I want to point out that we've shown a little bit of data from our scientific exploration of INS-1201 that's in animals, but really does, I think, show you why we think administering the drug into the spinal fluid, it will be effective and will be an advantage for us. We also discussed a little bit about the ASCEND clinical trial, which is ongoing and is really helping us understand how that dosing will work to enable larger studies going forward. Finally, I want to thank you all for your attention, and especially thank the families who put a little bit of faith and trust in us and tried something that might maybe sounded to them a little bit counterintuitive at first, but hopefully the results will speak for themselves. So thank you very much.
Good job. Nice one. That's good. That puts an end to what we're doing with gene delivery. We're going to move on, as I've mentioned earlier on this afternoon, to gene editing. This is the first time that we'll get a presentation. I think it's Gary Owens coming up now, is it? It is. And I'll let you kick it off from here. Thank you. Good, Gary.
Good afternoon. I'm Gary Owens. I'm the Duchenne lead for our program at Precision Biosciences. I'm also a scientist, and I've had the privilege of working on this program since it was on the whiteboard. So super excited to see it get to the point where it is today. I just want to thank the Cure Duchenne for having us here to talk about our early investigational drug. All of you for hanging around after a really good lunch to hear some science. And later on, we have a booth. If you want to have any questions, we'd love to chat more about some of the stuff we're talking about. So if you don't remember anything about what I say in the next 7.5 minutes, I want you to remember that this is a uniquely different approach where we're targeting the root cause of this disease at the DNA level. PBGene-DMD is the name of our therapy, and it's designed to excise the hotspot region of the dystrophin gene. So this is exons 45 to 55. The whole goal here is to permanently edit those cells so that the patient's body makes a near full-length dystrophin. And restoring the ready frame, restoring function is the goal. We have several reasons to be excited about this program. The first is there's a lot of exciting science that's been talked about today, and I love that we're sharing collectively, and so we can learn from one another. I think that's needed. But there's a huge unmet need because this is a really complicated condition. There's mutations across this giant gene, and there's not a one-size-fit-all therapy out there, and so I think we need to work together. I think PBGene-DMD is uniquely positioned to be very impactful in this therapeutic area because of our ARCUS technology. ARCUS is a meganuclease editor. You may have heard of CRISPR, but you haven't heard of ARCUS. ARCUS is different. It's very different fundamentally than CRISPR. ARCUS is a single component protein. It doesn't require guide RNAs, and it's actually really small, and we use protein engineering to modify this algae protein that's evolved to insert itself into genomes. We also have a very robust preclinical data package. I'll give you a glimpse of this when we get to it, but we have numerous studies in humanized disease mouse models that give us a picture of what potentially could happen in patients. In addition to that, we've spent a really long time talking to all of you, talking to top clinicians and regulatory folks to design a clinical protocol that is as safe as we can, robust, and will give us the results, hopefully, to show success and move forward in the clinic. So this is a cartoon that you saw a little bit ago. This really shows our therapeutic approach. And our approach here is we actually deliver our ARCUS nucleases via AAV systemically. And you can see in the cartoon here, we have two different ARCUSes, and they're designed to cleave different parts of the genome. And so we target the introns between exons 44 and 45, and 55 and 56. And when we do that, the ARCUS nuclease precisely cuts that in a sticky way. That region gets excised, and then the cell's own machinery stitches it back together. And now what you have is an in-frame genome, exons 44 with an intron to 56. So with this, it generates a now in-frame, near full-length dystrophin. The cool part about excising this hotspot, at least with our therapy, is 60% of all the mutations that are associated with Duchenne are in this region. And so it has broad applicability to the patient population. When we do this, we create what we're calling a near full-length dystrophin. And so we've got this cartoon here that shows full-length dystrophin at the top, and then the dystrophin that would be produced via the edit using the ARCUS nucleases. It's approximately 80% of the length of full-length dystrophin, compared to some of the smaller protein replacement therapies out there that are roughly about 35%. And there's growing talk in the field, the bigger dystrophin protein that you can deliver, the better. It has more function, it retains some of the areas that interact with nNOS and other complex cellular processes, but also the stability of the protein should hang around a lot longer. And that's really important. Pivoting a little bit, I want to kind of differentiate us as well, because we are an AAV-delivered therapy, but we're a different AAV-delivered therapy. We're delivering the ARCUS nuclease to do the editing off of our AAV, as opposed to delivering a transgene that makes the microdystrophin protein or some other protein replacement. Why is that different? Well, over time, potentially, the transgene can be silenced or diluted with growth. PBGene-DMD does not rely on the persistence of that transgene to be there. Again, we're editing at the root cause. All we need to do is to deliver the ARCUS to the cell. The ARCUS protein gets into the nucleus and makes the edit, and then the patient's own body makes the dystrophin protein. So in this case, in our case, the AAV is the truck that delivers the cargo. And the cargo does the work, and then the patient's body takes over. I mentioned robust preclinical data, and this is just a snapshot of a number of different studies that we've done looking at some functional improvements and other protein expressions that we've seen in a humanized disease model. On the left, you see restoration of dystrophin in the calf. This is representative of what we see in all the skeletal muscles in all the studies that we've done in this model. So we can take this model that doesn't produce dystrophin, deliver our ARCUS nucleases, restore it in those nuclei, and we see protein expression. And in fact, we see protein expression over time. In addition, in some safety studies, we've looked at CK levels. Earlier this afternoon, you heard CK as a measurement of muscle injury. And we actually see CK go down in these models after treatment over time. And then lastly, we're making this protein. We say that it's functional. We actually put these mice in functional tests, and we saw improvement in force in these models over time. In fact, we saw improvement over untreated at 3 months, but then it actually increased at 9 months. And so it's incredibly encouraging to us. In addition, we've got data in this model where we have edited the muscle satellite cells, which will help with durability. So any muscle satellite cell that is edited, when it activates and generates a new myofiber, that myofiber is going to retain the edit and is going to produce this near full-length dystrophin. So all of that was done in the lab and preclinically, and now we're excited to announce our phase 1 clinical trial. So it's a phase 1/2 clinical trial with 2 parts. Our first part will be safety. Well, all of it's safety, but our first part will be first 3 patients dosed. We'll look at the safety of that, and then we'll go into expansion phase. Some interesting things that we've decided to do with this trial is we're not going to do a dose escalation. We talked to a lot of you, and we know that parents and families don't want to be the first person on a low dose. And so we're going in at a safe, efficacious dose in these patient populations. Our primary endpoints here will be safety, obviously, and then at the secondary endpoint, we'll look at dystrophin expression. But we're also going to do some exploratory endpoints, many of the endpoints that have been shown here earlier today, so I won't dive into those much, but we want to get a relationship between this near full-length dystrophin and some sort of functional output. In addition, we are really keen on safety, as all of us should be, and we're employing a very robust immunosuppression regime as well as safety monitoring. Our inclusion criteria are males, 2 to 7 ambulatory, and they have to have a mutation within exons 45 and 55. It can be a deletion, duplication, nonsense. As long as it's in that region, then the patient should be eligible from a genetic standpoint. So, just kind of quickly, just going over why, what our goal here again is this is a single therapy with the goal of attacking the root cause of this disease to produce a near full-length dystrophin that we know has function in humans because there are humans walking around with this mutation. So we know this protein works. In all of our models to date, we've seen long-term muscle function retention. We've seen it increase. We've seen increase in muscle fibers expressing this dystrophin. So we feel, fairly good about where we're at. So we're thrilled just to let all of you know that we are currently approved for our IND. We are now activating and working with sites for startup and with the goal of getting into patients as soon as we can. This is Precision Biosciences. We're a small company from Durham, North Carolina. This is all of us on a warm August day. We're excited to be here and would love to talk to people later on today. Thank you.
Well done, nice job. It's a real scientific tour de force this afternoon, right? You're getting a bit of everything. We're going to change gears right now and talk about Tavard. This is a program that I talked about earlier on. We're moving into a space of AAV delivery with tRNA suppressors for reading through stop codons. Peter Amon's just joining us right now. All yours, Peter.
Hello. Good afternoon. As I said, I'm Peter Iman with Tevard Biosciences. I'd really like to thank the organizers for inviting us to speak here. It's a real honor, and particularly I'd like to call out Cure Duchenne. They've been a supporter of us over the years. We really couldn't do all the work we do without that kind of support. Just a quick background on the company, since we're somewhat new here. We're a Boston-based biotech. We're a fairly small, lean company, about 25 employees right now. We've been working in the Duchenne space for somewhere between 3 and 4 years at this point. It's just my disclosures. Mike gave a really nice introduction to how this technology works, so I'll try not to spend too much time on this slide. But I do want to call out that this is a fundamentally different approach from any of the others that you've heard about today. So we're using the cell's core translational machinery, and we're adding in one element, a suppressor tRNA, and that enables us to achieve read-through of nonsense mutations. So this therapeutic is, of course, mutation-specific. It's only applicable to people who have nonsense mutations, but it really is a mutation-specific gene-agnostic type of approach. And so we look at it as something that can be taken into Duchenne muscular dystrophy, but then rapidly expanded to any patient populations that have nonsense mutations in the muscular dystrophy space and far beyond that as well. So what we're doing in very simple terms, like I said, we're taking a transfer RNA and we're converting it into what's called a suppressor tRNA. This is a tRNA where we've changed the anticodon so that rather than recognizing a coding codon, it's now recognizing one of the 3 stop codons, and it's specifically recognizing a nonsense mutation, not native stop codons. And what this means is that for patients with nonsense mutations, the suppressor tRNA can actually read through that nonsense mutation. It can put back the amino acid that's supposed to go there, and most importantly, it enables production of the full-length protein rather than the truncated protein product. And I'll just mention one of the attractive features of this, in addition to it being sort of a broadly applicable platform. tRNAs are incredibly small, so even though we're targeting a fairly large gene like dystrophin, the actual payload that we're delivering is less than 100 base pairs in length, so it easily fits into an AAV vector. And again, this will give restoration of the full-length dystrophin protein, indistinguishable from the wild-type protein in patients with nonsense mutations. I probably don't need to convince anyone here, as we heard before, the larger the dystrophin protein that you can restore, the better. And we really believe this is one of the only options that will allow restoration of complete, full-length, 100% dystrophin protein in patient populations with nonsense mutations. So just a little bit of background on what we do. Like I said, making a tRNA into a suppressor tRNA probably sounds simple on paper. It's a little bit more complicated than that in practice. We are using AAVs to deliver our suppressor tRNAs. And what we've found is that if you just start with a generic tRNA and convert it into a suppressor, it'll read through nonsense mutations, but very inefficiently. So that's what we're calling our generation 1 suppressor tRNAs. And you can see that over the past 3 to 4 years, we've done quite a bit of screening to try and optimize this. So the plot on the right-hand side there is from the mouse model of dystrophin. It's the standard MDX mouse model. And we're initially screening doing IM injections. And so you can see when we started out, we were down around 2%. Not very impressive. So we did a bunch of screening. We added second site mutations to different regions of the tRNA to try and improve its efficiency. And we wound up with what we're calling Gen 2, generation 2 suppressors on this slide, bumped up to about 30% rescue. That was actually good enough that, as you'll see, we took these in vivo systemically by IV delivery. But we really felt like we could do better, so we've done a few subsequent rounds of screening since then. And you can see that with each round, we learn new things about suppressor tRNA biology that informs the next round of screening. And with what we're calling our generation 4 suppressors at this point, with the IM injection route, we're up to 100% rescue of dystrophin in these mice, going from 0%. So here is just a little bit more detail on one of our generation 2 suppressor tRNAs. Again, this slide is IM injection, so we're delivering a pretty good dose. And we're getting about the most rescue you could get with this particular suppressor. So you can see we're up around 20-30% dystrophin rescue in the muscles. And if you look by immunohistochemistry, you can see we're really restoring back dystrophin protein, and it's localized to the cell walls, the muscle fiber walls, exactly the way you would expect. But of course, for any clinical therapy, it's going to need to go systemically. So what does it look like in mice if we take one of these suppressors and do IV administration? What you can see here is that if we look at just full-length dystrophin as one of our readouts over on the left-hand panels, we're getting good restoration of the dystrophin protein in multiple muscle groups. I'm showing you gastrocnemius, quadriceps. We really see this in every muscle group we look at, from skeletal muscles, diaphragm, to cardiac muscle. And again, since the suppressor tRNA is only acting on the nonsense mutation of the dystrophin gene, it'll restore dystrophin in cells where it needs to be, and it's effectively inert in other tissues. We can also look at various markers of muscle injury in these mice, and we see a nice downward trend that correlates with restoration of the full-length dystrophin protein. And then what does this mean functionally? So we're really excited to see how performance improves in these mice in some of the exercises that we run them through. So grip strength is pretty much exactly what you'd expect. The mice hang on to a device that measures how much strength their muscles can exert. You can see in white, that's grip strength for wild-type mice. That's what we'd like to be at. The gray bars are untreated mice, and mice that were treated with a control AAV that doesn't include a suppressor tRNA. They're clearly reduced from wild-type, and we actually get 100% restoration of grip strength as a function. We can also look at rotarod, a little bit more complex behavioral test where the mice run on a rod for as long as they can as we speed it up, and we measure how long they're able to stay on. Wire hang is another one. In all cases, we see really nice restoration of function, pretty much up to 100%. And I'll say that this is happening very quickly after administration. With the Generation 2 suppressors that I was mentioning, we were getting this sometime between 6 and 12 weeks after injection. With the new Generation 3 and Generation 4s, we've actually seen it as soon as 3 weeks after injection. So everything that I've been showing you so far is from a suppressor tRNA that targets a TAA stop codon. Obviously, if we want to treat all patients with nonsense mutations, we need a couple more suppressors at minimum. We need ones that are going to target TGA and TAG stops. Ideally, we would like to have these in different flavors so they can put back the amino acid that's supposed to go there. So we're working to expand this quite a bit. What you can see on this slide, we've just taken our TAA suppressor that I was showing in one of the previous slides, and we've changed that anticodon on the optimized suppressor so that now it can target either a TGA nonsense mutation or a TAG nonsense mutation. And we were really reassured to see that these improvements that we engineered into the initial one really translate across the board. And so we feel like we've got a good path forward here to expand this platform into all different classes of nonsense mutations. And then I just wanted to put this up there to give a little bit of a sense of where we see this type of therapy fitting into the landscape of different therapeutics and addressing unmet patient needs. And I guess I probably picked the wrong comparison here. This is the microdystrophin talk, but I just wanted to show how it compares with exon skippers. And what I really wanted to call out here is that a lot of the exon skippers are targeting, obviously, very specific exons. Nonsense mutations, on the other hand, are spread pretty much throughout the length of the dystrophin protein. So the plot you see on the bottom here is essentially the dystrophin protein, every exon indicating prevalence of different nonsense mutations on it. You can see that a very small fraction of these are actually amenable to an exon skipping technology. So we really see this as being a way to expand treatment options for patients who are maybe not amenable to an exon skipping type of approach. It's one more piece of the pie, as you can see. So just to sort of wrap up, we see our technology as being hopefully a best-in-class therapy for patients who have nonsense mutations. The work that I've shown you so far is all preclinical. We feel like we've identified very good suppressors for certain classes, so I'm hoping that we'll be heading into the clinic very soon. But we do see with these that we really are getting very significant restoration in multiple functional endpoints, and we've seen no indications of safety concerns so far in mouse models and some preliminary studies in larger animals as well. And we really feel like this type of therapy has the potential to treat a majority of DMD patients with nonsense mutations. And we've started to build up a fairly extensive toolkit for treating different types of nonsense mutations. I think with something like dystrophin, there's probably actually a fairly high tolerance for what I would call miscoding correction. So I would suspect with just 3 different suppressors, one for a TAA, one for a TAG, and one for a TAG, we'd actually have a therapy that's relevant for any patient with a nonsense mutation in dystrophin. So with that, I'd just like to thank the entire team at Tvard, and of course, I'd like to thank once again the organizers of this conference.
Thank you, appreciate it. Thank you, that was Heavy Science. We're going to change gear one last time. I'm going to invite Barry onto the stage, and we're going to, I think you're talking about immunosuppression, et cetera, et cetera?
Yeah, thanks, Mike. Really
Okay.
appreciate the opportunity to speak to you all today. I'm from the University of Florida, which is right up the road. If none of you have been to Gainesville, it's just about 2 hours from here. Our campus is actually where AAV vectors were first discovered. And a lot of the progress you've heard today is based on a paper we published in 1997. So we really need to keep up the progress because I think we're close. And what I want to do today is talk to you a little bit about a holistic view that we've taken towards the notion of gene transfer in Duchenne, particularly focusing on some of the immune management problems that have arisen with more widespread use of gene therapy vectors. So this care guideline was an important step in really understanding all the details of what happens when a patient becomes eligible for gene therapy, what happens after they receive gene therapy, and how we prepare for long-term follow-up. This was a collaborative effort, listening to patients with 45 experts in the field to give their best opinion on this paper that was published late last year. We've taken a unique approach in Florida. So there are about 22 million people in this state and about 250,000 births per year. Representative Adam Anderson, at the time Duchenne newborn screening was not yet approved in any state, began a progress towards a system that would allow us to do whole genome sequencing in an opt-in program called the Florida Sunshine Genetics Act. Concurrent with that, Duchenne newborn screening specifically was also added in state law last year. And so, in fact, as a result of this program, we intend to do a genetic-based screening following CK pre-screening. In states that are doing CK screening and confirmation, families that don't have Duchenne may learn that they need to have confirmational DNA testing and be suspicious about a muscle disease that may never come to fruition because they had a birth injury. So we think this genetic-based testing is going to really be an important way to focus on those that actually have Duchenne and need specific follow-up. And genetic testing is really the cornerstone of many of the treatments you've heard about. Mutation-specific therapies like exon skipping or eligibility for gene therapy products where some exons, some mutations are excluded. And so really, I think we're zooming out now on the notion that earlier treatment, as with many inherited diseases, will be beneficial. And that, I think we've learned in Duchenne from some of the great work that's done on natural history, as well as patients treated with gene therapy across the age spectrum. We know that there's a point at which muscle regeneration starts to influence the structure of the muscle such that there may be an irreversible change. So this is what's driving the potential for early treatment. In the context of cardiomyopathy, we have so much redundant function that this is not a concern really early in life, and we do want to make sure that those that could benefit from gene therapy treatments, even those that are non-ambulant, would have access to treatments that can make a difference in heart function. So this is the complex dystrophin gene. This figure is from a review we published late last year, and you can see how much of dystrophin is missing from these various constructs that are based on a single gene therapy vector. You heard a little bit about how gene editing, precise gene editing, or stop codon suppression could result in longer dystrophins. But there's one additional way in which that can occur, and I'll explain that in a moment. We also wanted to focus on many of the things that exclude patients from access to gene therapy and how to make gene therapy products safer. I think there's been a lot of progress now on improving critical quality attributes of gene therapy products, and that has actually had a substantial impact on lowering the adverse event rate, which, as has been said, is really a class effect of gene therapy vectors. But one of the most important things we can do is improve the efficiency of the delivery truck or the capsid that targets muscle cells. And importantly, we found recently that we can design capsids that not only target the musculature, but can also target the brain. And about a third of patients have significant behavioral problems and sometimes even cognitive problems because of the missing portion of dystrophin that is essential in brain function. We want to optimize the sequences that drive the expression, and that requires, again, some unique considerations, particularly if one wants to target the central nervous system and the musculature and the heart. We can reduce immunogenicity of gene therapy vectors by changing the codon utilization, and that's another way of improving efficiency. And as I said, manufacturing is a really important part of improving safety. So to develop new capsids, we presented last week at the American Society of Gene Therapy the work done at University of Florida to do multi-species evolution of an AAV capsid that has been selected for muscle and heart targeting. This has about a 20-fold efficiency over its parental strain, and the scaffold that it's built on was designed specifically to not traffic to the liver. So I'm going to show you some of the data with those capsids and how they pertain to two publications which were published in January of 24. Jeff Chamberlain, you heard about, has really been a pioneer in developing novel microdystrophins. His group, led by Haitham Tashford, also showed that AAV can be used in a multi-vector format to make a near full-length dystrophin. At the same time, a similar paper was published by the group of Renzi Han at Indiana University, and we've been longtime collaborators with Roland Herzog and Renzi to bring this forward to the clinic. The concept requires the ability to make AAV from two parts using a technique that bacteria use to make larger proteins called protein transplicing. Protein transplicing uses two inteins that are designed to be on one and the other end of each of the AAVs. After those proteins are made, they join the protein together to make, in this case, the mutation that we've focused on, the in-frame mutation, makes a 280 kilodalton form of dystrophin, which is about 2/3 of full-length dystrophin. It's the same schematic shown relative to Elevidys, and you can see this central rod domain deletion, as was mentioned earlier, is not associated with any clinically significant disease in Becker patients with central rod domain deletions. And shown in the right panel is the representation of a number of these capsid variants, making the near full-length dystrophin in a level of efficiency that we're quite surprised is actually can exceed that of even microdystrophin. Partly because once this larger protein is stabilized in the membrane, it stays there for the life of the cell. In the center there, you see in the red the same type of protein, attempting to make that with AAV RH74, which is how Elevidys is made. The efficiency is not sufficient to make the protein compared to these novel capsids. So we actually today submitted a very large program grant to the federal government, to the NIH, to study all aspects of the immunology and the delivery of near full-length and full-length dystrophin. And one important thing about this collaboration with the group in Indiana is this is a finding we had both for how to be inclusive of all patients with all even prior exposure to AAV using immune modulation technologies that we have developed over the last 10 years. A really important finding from this that would enable all mutations to be included is that in a separate publication that we've shown that expressing a tiny amount of dystrophin where in the hepatocytes where it doesn't actually have any function other than to create hepatic-specific immune tolerance or antigen-specific immune tolerance, that enables any mutation to be eligible for gene therapy, unlike some of the circumstances where some mutations are excluded currently. And these will all utilize these new capsid technologies that we've developed. So if I can take a minute to imagine what would be the perfect scheme for receiving such a therapy, the target product profile is an approach that the FDA uses to identify the key features of a therapeutic plan. So we think it's important to have newborn screening. This would allow for early interventions with some of the adjunctive therapies that are being developed now, such as sevasemten, HDAC inhibitors, because very early gene therapy at age 0 is going to be subject to some degree of loss of the transgene because of growth. Sometime in the early ambulant phase, the amount of injury increases substantially. So we do think that in that early childhood period, with the intention of being able to retreat those patients at a later age is an appropriate way to go. And that will sustain expression lifelong, and this requires having the technology for redosing, which is of course relevant to all those pioneers that adopted the current therapies very early on, and it may be more than 5 years after treatment where we know the effect begins to decline. I mentioned the CNS disorder. We think that's going to be an appropriate way to differentiate this approach from others. And we do think that being very focused on the cardiomyopathy will be important to those non-ambulant patients. So we're not focusing only on those early identified patients, but also those who may have developed cardiomyopathy where there's still an opportunity to intervene. And then really importantly, we want to make this available ultimately worldwide through some innovations in manufacturing that are intended to dramatically lower the cost of AAV products so that they can be available to everyone. Our institution has actually coalesced a lot of these efforts into the UF Health Center for Advanced Therapeutics. This is where all our commercial products are delivered. We really appreciate this partnership with UF Health because we're really trying to make this available to everyone statewide and differentiate what we're doing in Florida from other places. And I always want to say thanks to the team that makes this possible. We just celebrated an event where we identified our 100th patient treated with gene therapy, so it's been really valuable to have the support of our funding agencies and this team that has made it all possible. So thank you for being here, and I'm around the rest of the weekend to answer any questions you have. Thanks.
Thank you, Barry. So that concludes this session. I want to thank everyone who came up and talked. There was like quite a wide range of topics on here, and I know it'll touch quite many of you. You'll see what's in the clinic, what's been approved, and what's coming next. So just join me in thanking the presenters one last time. Thank you all.